Let's go ahead and get started. Any questions before we get rolling today? You guys in there?
Good morning.
Good morning. Excellent. All right, let's talk game plan. Today is Wednesday, September 9th, 2020. We are going to finish up our last bit of the lecture. We have to talk one more major physiological process. That's going to be protein synthesis. Then we will have time for preparation for the exam. That is going to include two things: practice lab exam, which you guys are going to take. Again, there are no points on this. This is just a chance for you guys to be able to gauge yourselves on how you've been preparing for this, and then we will talk about that. And then I'm hoping at the end of that, we will have time for a question and answer review. Again, review isn't me standing up here and telling you what I think is important. That is what I do every day in class. The review is a chance for you to ask questions about things you are unclear about, and we will discuss them and try to help to make sure that you understand those concepts. And remember, as I've said before, if you don't ask questions, I assume you have mastered the material and I make the test harder. All right, excellent. All of this leading up to Monday, where we have our first lab and lecture exam. Again, you may take those exams in any order that you want, but they must be completed during the class time. From 8 a.m. to 12:35 p.m. and again must be completed during that time. So that means if you start the lab exam at noon, you're only going to have 35 minutes to complete it. Again, there are time limits on them, so you don't have the entire time, but you must complete them during those times. And again, it's using the Proctorio protocols, the same protocols that were used for the syllabus quiz, the same one for the chemistry quiz. So as long as you took those two, or at least one of those two quizzes, you know that your system works with the Proctorio and it should not be a problem. And then Wednesday, we start our tissues and integumentary system. I will warn you, that is a tremendous amount of histology. We're going to do a lot of microscope work, and we don't have microscopes in front of us. So it is going to be a bit challenging. Anytime there are easy ways to learn stuff, I try to give you any of the cheats and tricks and mnemonics that I have to learn things. But with the tissues, with histology, really it's kind of about brute force memorization, familiarizing yourself with that stuff. So that's why having a good histology atlas is going to be something that if you don't already have, hopefully you will get by then. All right, so that is the game plan. Any questions on that before we get started? All right, excellent. Stunned silence, what I love first thing in the morning. All right, let us move on to protein synthesis. So there is this general rule or concept that we talk about in biology. As we talked about, our DNA, as we mentioned, contains genes. And the vast, vast, vast majority of the genes in our DNA code for information for making proteins. And so there's this general concept that one gene produces one protein. However, if you recall, we've talked about the structural classification of proteins, and we know that most proteins are made up of individual polypeptide subunits. So really, it is more accurate to say that one gene gives us the code and the information for making one polypeptide. So if you remember when we talked about, where's my annotation? There it is. When we talked about, oh, hey, Joed, I hope everything is okay. When we talked about hemoglobin, for instance, we talked about it being two subunits, two alpha subunits. So one gene would code the information for making two copies of that. And then a second gene would give us the code to make the beta subunit. And we put those two alpha and those two beta together, and we have that one fully functional hemoglobin protein. Right, so one gene basically codes for the information for making one polypeptide, and we put those polypeptides together in our Golgi apparatus to make functional proteins. Now, the process of making that polypeptide, of making that functional protein, basically requires two steps. The first is transcription. Right, transcription is the part of going from our stable, locked up in its nucleus DNA, to that temporary movable copy that is our RNA, right? If you think about it, back in ancient times, right, there were things called books, right? And books had valuable pieces of information. So they were typically locked up in places like churches or castles or things along those lines. And you couldn't just show up there with a library card and be able to check that piece of information out. Sounds good. Check that piece of information out. Instead, you would have to go to the church, you'd have to go to the king or whoever it was and ask them for that information. And if they deemed you worthy, right, would they even then give you necessarily the book and let you take it out of the castle? You guys can't hear me, right? This is the part where you guys respond. Would they let you check out the book? No. Excellent. No, Haley, you're very, very confident, but no. Typically in those ancient times, they wouldn't allow you to do that. Oh, okay, got it. No, what they would do is they would make a copy of the piece of information that you want, and then you would be able to take that. And of course, who were the people who did that type of work, made the copies of the information?
Scribes.
Yeah, they were the scribes, and that's where the term transcription comes from. You know, one of the ironies of the scribes is most scribes were illiterate, right? Because knowledge was power. So all a scribe would do was he would see a word, and then he would copy that word on a separate piece of paper without really even knowing what that piece of information was, right? And so that's what they would do. They would take a copy of that piece of information, they would make a copy of it, and that is what you would leave the castle with. And even if you go back to medieval times and do that and get permission and get the information, it probably wouldn't do you a whole lot of good. Because if you got that piece of information or a copy of that piece of information, right, most likely it was in Latin. And how many people here speak Latin? Anyone? Bueller? No, no one here speaks Latin. Exactly. So once you got that copy with that piece of information, what would you need to do?
Translator.
Exactly. You would need to translate it. And that is the second step in the process, translation. Translation is where so really transcription, if you think about it, is where we make a copy of the information. And translation is where we are changing the language of the information. We are changing the language from nucleic acids, the language of our DNA and our RNA, into the language of proteins. And the language of proteins is amino acids. So we have to change that information as well. And as we mentioned, transcription occurs in the nucleus. Where does translation take place? I'll wait for an answer. Cytoplasm. Okay, we get partial credit for that. We can be a little bit more specific than cytoplasm. If only we had a test in 5 days where I was responsible for the organelles of a cell and knowing what their functions were. Excellent. I like ribosome. Ribosome gets us a lot closer. Ribosome is an acceptable answer. I certainly would accept that on the exam. However, as we know, where are most ribosomes located? Now, you're definitely not going to get away with RER on the exam. That's a spelling error, right? But what does RER stand for? There we go. Rough endoplasmic reticulum. Excellent. So that is where that occurs. The ribosomes definitely are what do the work, absolutely. But since most of them are located on the rough endoplasmic reticulum, I would accept that as a correct answer as well. All right, excellent. So, and again, of course, we've done it with the words. Here's the pretty picture that kind of illustrates that process here as well. All righty. Let's talk about these processes. Oh, and as I mentioned, I think at the end of last class, but this is probably a good place to remind ourselves of that again. Anytime we have two related processes like this, how many possible essay questions does that lend itself to?
3.
3. There you go. Excellent. What are the 3 questions?
Explain each one and then compare them.
There you go. Describe the process of transcription, describe the process of translation, compare the two. How are they different? How are they similar? Exactly. Excellent. All right, so let's do that. Starting first with transcription. Again, as we mentioned, this occurs in the nucleus. And our job is to make a copy of the genetic information. We are still keeping it in the same language. We are using the language of nucleic acids, but we are changing it from a temporary version, probably from a permanent version to a temporary version of that information, right? We're just changing the dialect, right? We're going from California to Texas, so where we say hi and they say howdy, right? It's the same language, just different dialects of it. Now, remember, as we talked about, this requires a massive number of proteins, mostly enzymes, which of course are proteins, but we'll emphasize that there, nucleic acids, and et cetera, and a lot of other things. And remember, all of these things clump around a small section of the genetic material, a small section of the chromatin. But so much stuff condenses around it that where this process of transcription takes place, it becomes so dense, it becomes a structure that we can actually see with a light microscope. So anywhere in the nucleus where transcription takes place, we see a structure. And what did we call that structure? Where that transcription is taking place? Excellent, a nucleolus. Excellent. So that nucleolus is where all of those things are taking place. And again, this requires dozens and dozens of enzymes, all sorts of other things. But not surprisingly, the most important enzyme in this process is RNA polymerase. Now, if you think about it, RNA polymerase sounds very similar to DNA polymerase. Where did we use DNA polymerase again? Where did we use DNA polymerase?
DMA, when we were splitting the DNA.
Excellent. Perfect. When we were doing the replication, right? A replication of the DNA. And we did that during S phase, right? In preparation for division. If you remember, that DNA polymerase basically pulled apart the two strands, and then basically using our complementary base pair rules, it basically made new strands of DNA to replace the ones that were there before. And RNA polymerase is going to work in a very similar way. We're going to open up our DNA and we're going to read a strand of the DNA, and using base pair rules, we're going to make a copy of that, but we're going to make that copy in RNA. And that is the one trick in this process. We make a copy of the DNA with RNA. And there's one big trick to this process. Oops, got to clear my writing. Remember, our DNA polymerase took advantage of the number of hydrogen bonds that our nitrogen bases form, where we have these complementary base pair rules. And that's a big fancy word, so let's write that down. Where adenine only can bind with thymine, guanine can only bind with cytosine. But there was a difference between DNA and RNA. There's actually two differences between DNA and RNA. The first is which sugar they use in their, exactly, there you go, Haley. The first of the differences is the sugar that they use. DNA uses deoxyribose, whereas RNA uses ribose. But the other big difference comes in the form of this thymine. Remember, our DNA uses adenine, thymine, guanine, and cytosine, whereas RNA uses adenine, uses guanine, uses cytosine, but with RNA, Instead of thymine, it uses uracil. So basically the big difference is anywhere you would normally put a thymine, you are going to put a uracil there instead. All right, so if we had a strand of DNA, let's go ahead and make a strand of DNA here. So again, let's go with C, G, T, A, right? And this was our strand of DNA. If we wanted to make a strand of RNA, using the complementary pairs, what would go here? What would the first one be? What would be complementary to C?
G.
G, excellent. What would be next?
C.
What would be next?
T.
T, T goes with T?
A, sorry.
A, excellent. And what would go with the A? U, excellent. So that would be the complementary strand of RNA that we would make based on the DNA strand. Right, we use the same complementary base pair rules. The only difference is that anywhere we would normally put thymine, we're going to put uracil as well. All right, and like I said, the sugar backbones are different for these as well. And so I tried to emphasize that with color. All right, and that's pretty much how this works. Right, so let's talk about transcription. Let's do it first pretty on the board. Actually, let's just do it this way. This works. All right, so we have one bit of an issue. Actually, I do want to use a different board. So part of the issue is we have a massive amount of DNA. And that DNA, as we mentioned, codes for genes. How many? How many genes are actually coded on all of our genetic material? Lots. I like the term lots. A lot works. Yeah, sure. There are tens of thousands of genes. All right, whoops. So I spell it right. Tens of thousands of genes found on our DNA, all on those 46 chromosomes, right, that we have. But what's remarkable about that is the genes only make up about 1.5% of all of the DNA. So that means the vast majority of the DNA that we have is what is commonly referred to as junk DNA, a bunch of repetitive patterns of sequences of nucleotides that for the longest time they felt didn't have any significant meaning. Now, there is a new field of study called epigenetics, which is looking at some of these large massive sequences, and there may be some implications and roles, but that's far beyond what we need to worry about in this class. Our job right now is to make a protein. So here's our issue. If I just randomly closed my eyes and threw a dart at the DNA, what are the chances that I'm going to hit a gene, let alone the gene I want? But what is the chances of me just hitting a gene? It's not very good. So I can't just randomly decide that I want to make a gene. I need to be able to find the gene that I'm looking for. And so the way this occurs, the first step of this process is that we have to find what is called a promoter region. This finding of a promoter region is typically a special sequence of nucleotides, something like AAA, TTT, or something along those lines. And that tells our RNA polymerase that this is the binding site where we want to start. So let's make a sequence of DNA, AAA, TTT, CG, ATC, ATCG, ATC, and so on and so forth. Okay, S says it doesn't work, but you get the idea. Excellent. This strand of DNA right here would be what we would call, oops, no, I don't want to do that. Let's leave that black. What I want to do is this here, and make that red. this is our template region, or template strand, sorry, is what I should say. This is the strand where we have the information that codes for the particular gene. But as we know, DNA strands don't hang out by themselves. There is going to be a complementary strand to that. There's always that complementary strand. And as it turns out, that complementary strand is also sometimes referred to as the coding strand. This does not contain the information for the gene we want. This just happens to be the one that is the complementary strand. And what would that complementary strand be? Give me the sequence of nucleic acids that would be on this complementary strand.
C, T, T,
Yep.
A,
A. Then what?
G, C, T, A, G, T, A, G, C, T, A, G.
There you go, dot, dot, dot. All right, excellent. Perfect. So this is our starting point. This is our strand of DNA that has that complementary strand and has that template strand. What's going to happen is our RNA polymerase is going to find this promoter region. And when it finds this promoter region with the help of other enzymes, it is going to separate those two strands. Remember, it's only hydrogen bonds that are holding it together. Then what it is going to do is it is going to attach to this point, start to separate it, and it is going to move down the line. And as it moves down the line, it is going to make a copy, do green, of the DNA material, the information that is here, but using RNA nucleotides. So if we were to use an RNA nucleotide, of course, the first one would be G, oops, G, and then would be C, and then what would we have? What would the next one be? U, excellent, uracil, we'd use that uracil, and then an adenine, and then a guanine, and then another uracil, and then an adenine, and so on and so forth, down the line it would go, right? So the first thing it has to do is attach. Attachment occurs at that promoter region. Then we have this elongation, where we are using this, I shouldn't have been blue, that should have been green, I don't know why that changed. We are using our complementary base pair rules to go down the line, making a copy of the information. But again, here we are now using RNA instead of using DNA. Where this is occurring, we get this nice kind of interesting area here where we get this RNA-DNA matrix that is forming, where we have the two strands together over a set period of time. And again, this elongation continues down the entire length of the DNA. But does it truly go the entire length and just read all the way down to the end of whatever that 46th chromosome would be? No, eventually it's going to reach a termination sequence. And that termination sequence could be something like AT, 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 or something along those lines. And at that point, it is going to let go. All right, let's look at this and talk about this with the pretty words and the pretty pictures. So again, this RNA polymerase unwinds and copies the DNA, makes a copy of it into RNA, and it has three key steps in this process of transcription. The first is initiation. That is, again, where it binds to that promoter region, and again, that promoter region is a special sequence of nucleic acids. Oops, I don't know why everything's in capitals. nucleic acids. And like I said, an example of this, and this is just one example, there are many of them, but AAATTT. You don't have to memorize that the same way you have to memorize some of the other things that we'll talk about, but that's just an example of that. And that is going to tell us our starting point. That's that DART finding that 1.5%, finding that gene that we need. Once it initiates, then it starts to unwind. But notice here that one of the big differences between the two is here it only temporarily unwinds the DNA. Notice at the end of this process, the DNA is unchanged by this process. Unlike DNA replication, where we tear the two strands apart and use them as templates for making new strands of DNA, and suddenly we have two strands of DNA where we had one before. Here our DNA is completely unchanged by this process. So it pries it apart temporarily and starts working its way down using our complementary base pair rules for this elongation step. In this elongation step, as we can see, we are temporarily separating the DNA. The DNA is unchanged, but we're using our complementary base pair rules to make a big strand. And I hate that your book does this. I'm going to cheat. I need this. It makes a strand of RNA. So here we have riboses and phosphates forming a backbone, and we have a strand of nitrogen bases, A's, U's, C's, and G's, making a single strand of RNA. And again, we use those same complementary base pair rules we've been using again and again. Now these illustrations are very simple, basic ones, but here I have the pretty picture from your textbook. Here you will see the pretty picture of your textbook where we see this DNA RNA hybrid that we were talking about. Notice here is where we're unwinding the DNA, right here. Here is where it winds back together. So again, it is unchanged. Notice we're only interested in the information on one of the two strands. So this strand right here is our template strand. This is the one that has the information we are interested in. Whereas the other one, the complementary strand, is also known as the coding strand. And here, anytime we have an A, we put a U. Every time we have a C, we put a G, and so on and so forth. So we have this area where we have both DNA and RNA together inside of our RNA polymerase, and we call this region the DNA-RNA hybrid region. And like I said, eventually, it doesn't just go down to the end of the DNA and just fall off the end. Eventually, it reaches a special sequence known as a termination signal. And once it reaches that termination signal, the RNA polymerase lets go of the DNA. Our DNA is completely unchanged by this process. And we have a long sequence of RNA. Now, we have made this transcript. However, this strand, and again, this is where, again, this is a simple illustration that kind of doesn't emphasize the things. It is not mRNA yet. Any idea what mRNA stands for? What does the M stand for? Messenger, there you go. Messenger RNA is our goal of this process. This goal of this process is to make that temporary copy of the genetic information that can leave the nucleus and allow us to make a protein. But at this point, what we have is just a completed strand of RNA. This strand of RNA must then be modified. By modified, what happens is we are going to cut sections of it out. And as we cut those sections of it out, we are then going to put those pieces back together. And once those pieces are put back together into a single continuous strand, then and only then do we have our messenger RNA. And the good news is once you've written out messenger RNA once on your essay question, you may then abbreviate it mRNA. So there is an additional step that this simple illustration doesn't show. We must modify that strand of RNA. And once we've modified that strand of RNA, then it becomes our messenger RNA. Once we have our messenger RNA, it leaves the nucleus, and where does it go again? Where is our messenger RNA going to go? Give you a hint, it's not to the store to buy milk. Excellent, to the rough endoplasmic reticulum. Perfect, to the rough endoplasmic reticulum. Excellent, and at that rough endoplasmic reticulum is where we are going to change the language. So that's the next issue. We need to then, once in step 2, we need to change the language. But we have a problem with changing the language, right? We, for the most part, use English in this class. And how many letters are there in the alphabet of English? 26, excellent. And we take those 26 letters and we switch them around, put them in different orders, and from that we give them meaning, right? For our nucleic acids, how many letters are there in their alphabet? Right, well, technically there is 5. Right, letters in their alphabet, but Adina's got the right idea. For DNA, there is 4, and for RNA, there is 4. So there's 4 in RNA: U, A, C, and G. But what about proteins? Proteins are made of amino acids. And how many letters are there in the amino acid alphabet? How many different amino acids are there that we put together in different combinations to make all of our proteins? 20. Excellent. Right? And again, do you have to memorize those 20? Know their functional groups? No, you don't need to. No, you don't. If you want to do that, you're welcome to take a biochemistry class. So no, I'm not going to make you do that. But we do still have this issue, right? Because we only have 4 letters in our nucleic acid alphabet, and they need to code for our 20 different amino acids when we're making a protein. So does that mean that U stands for 5 different amino acids and we just randomly select one of those to put them in? Is that going to be an efficient way to make proteins? No. So it doesn't stand for 1, it doesn't stand for 5. So how can we do that? How can we use nucleic acids to code for specific amino acids? How can we make this work? We can't, which is why we don't make proteins, which is why we're all really dead. All just figments of some giant turtle floating through space's imagination. There you go, exactly. We have to combine them together, right? So A can't equal a specific amino acid, but maybe AA could, right? To find out for sure, we could figure that out. It would be, instead of 4, it would be 4 squared. And what is 4 times 4? What does 4 squared equal? 16. So that's not going to be enough, absolutely. But luckily, as who was it, Haley pointed out, if instead we did something like AAA, that would be 4 cubed. So 4 Where's it? Times 4 times 4. And what does that equal? I'll wait if you have to take off your shoes and socks.
64.
There you go. It equals 64, which is more than enough combinations. So absolutely, what we are going to do is take sequences of three nucleic acids together, and those 3 amino acids together will code for a specific amino acid. There we get far more than we need. We're going to get some repetition, but it is going to work. And of course, we have to give a fancy name to that sequence of 3 nucleic acids, and that fancy name to the sequence of 3 amino acids is a codon. So we are going to use codons, and let's pretend I spelled sequence correctly. There we go. Is going to be the code that we're going to use for an amino acid. Now, any book worth its salt, and yours is one of them, has one of these fancy tables that show us exactly how this works, right? So I keep talking about these different examples. Here we go. For instance, if you had a U for the first one, a C for the second, and a C for the third, then that codon UCC codes for serine, right? CCC codes for proline, right? And so on and so forth through all of these. Now, do you necessarily have to memorize this chart? No, Allison seems pretty certain that the answer is no. And yes, absolutely. The answer is no. The good news is you don't have to memorize this, right? Do you need to be able to read it and know how it works? Absolutely, you have to know how it works. However, I do notice that there are 4 different codons that are highlighted with special colors. One of those happens to be AUG. AUG is a special codon. You do need to memorize it. And as someone has already pointed out, and the fact that it is green kind of should tell you that it is the start codon. And guess what you do at a start codon?
Start.
Yeah, there you go. You start. This is where we will start the making of the polypeptide. This is where we're going to start making our polypeptide. And not only is this the start, but if you look closely at our chart, AUG codon always codes for the amino acid methionine. And that you do need to know. That is the one amino acid whose codon you need to know, because AUG always codes for methionine. Notice also there are 3 more highlighted codons, UAA, UGA, and UAG. These are all red in color, so guess what those are? There we go, stop codons. And guess what you do at a stop codon? You stop, there you go. This is where we are going to stop translation. So our start codon is where we start translation, start making our polypeptide, or we can say this is where we start translation. And our stop codons are where we stop translation. Now notice, with our stop codons, do they also code for amino acids? Looking at the table, what do you think? No, exactly. So these do not code for amino acids. So then the question is, what do they code for? And the answer is, I will tell you that later. All right. Excellent. Questions on this? All right. So again, you do not have to memorize this chart. You have to be able to read a chart like this, but you do have to have the 4 codons that are highlighted memorized. You need to know the start codon. You need to know what amino acid it codes for. And you need to know the 3 stop codons. and what they do. All right, excellent. So this is some of the information that we're going to need with that messenger RNA to be able to make our amino acid sequence, this process of translation. All right, translation is when our messenger RNA is converted into, and again, notice here it says into a protein, but again, we can be a little more specific by saying this is where we code it into a polypeptide. Oops, why is that not working? And that polypeptide is going to then be sent to the Golgi apparatus, where it will be converted into a functional protein. But messenger RNA isn't the only molecule necessary for this process. It's not even the only RNA that is necessary for this process. There are two other types of RNA that are going to be needed for the process of translation. And those are transfer RNA and ribosomal RNA. So we need to talk about these two other forms of RNA as well. And again, once you've written out transfer RNA, you can abbreviate it lowercase t RNA. Once you've written out ribosomal RNA, you can abbreviate it lowercase r RNA. Or like as I said before, on an exam, if you want to have a key at the bottom or at the top where it says tRNA equals this and rRNA equals that and so forth, you can do that. Let's talk about the transfer RNA first. Transfer RNA is the smallest of the RNAs. It is just a single strand of RNA that is only about 80 nucleotides. It is really, really small. But the key to it is it has two main binding points. At one end, it is going to contain a specific amino acid that it is going to bind to and latch on to. And at the other end is going to be the code that is going to allow it to attach to our messenger RNA. Now, so far there's only one codon that codes for an amino acid we have to know. And that right now for our messenger RNA, as we know, is AUG. All right. AUG is our start codon. And what, there it is. What amino acid did we say that it bound to it? Or codes for, I should say. What amino acid does it code for? Methionine, excellent. Which, again, since we've written it a couple times already, I will just abbreviate MET. So we know that if our messenger RNA has AUG as its codon, then that means we want and need the methionine. Move that there, move that. Oh, gravy. This is so much easier when I have a board and crayons. All right, excellent. We need our transfer RNA to bring that specific amino acid. So here is our transfer RNA. It is bound to our amino acid. But we need a way to know which transfer RNA to bring in. And we do that with what is called an anticodon. The anticodon uses the complementary base pair rules to match up with the codon. So using our complementary base pair rules, what are the three nucleic acids that we would need here to make it equivalent to our, or using the complementary base pair rules would match it up with AUG? What would the first one be? What's complementary to A?
U.
U, excellent. What's complementary to U?
A.
And what's complementary to G?
C.
There you go. So what we would have here is that matching set. What this one here is, and let's make this small enough so I can fit it in. There and there. So here we have our codon on our messenger RNA, and here on our transfer RNA, we have an anticodon. So basically, our transfer RNA has two key sides to it. It has the base, where we have our anticodon that we're going to use to match up to the messenger RNA. And the second part is the binding site, where it holds on to a specific amino acid. And that amino acid is going to match whatever the codon is, so that codes for that specific piece of information. Now, I've done a truly amazing job drawing this, but much like proteins, RNA has structural organization to it. So notice here, we see the primary, or really two-dimensional sequence of this here, where we have, it's kind of this clover leaf shape to our transfer RNA, where we have these disulfide bonds going on. And again, notice we have our 2 points here. Up here is our attachment point for the amino acid. And here at the base of the clover is where we have those 3 specific nucleic acids that is going to be our anticodon. Now, this two-dimensional structure folds up into this elaborate-looking three-dimensional structure that we see here, which is pretty elaborate-looking, and we don't have to worry about memorizing the three-dimensional shape. But notice, symbolically, it is often drawn as a cylinder or a bent cylinder like the one we see here. For simplicity's sake, I will just draw it as a cylinder. And then again, it's going to have a binding site for an amino acid at one side, and on the other side, it is going to have an anticodon. And that anticodon would allow it to bind to a specific codon. And in this case, if the anticodon was CCC, what codon would that bind to? GGG, excellent, GGG. And do we know what amino acid that would bring then to the party? Yeah, exactly. We could look at the table and figure it out. I don't know it off the top of my head, but we could look at that table and we could figure out what GGG. Oh, is GGG for go for glycine? Perfect, excellent. So we could actually go there and figure it out. All right, excellent.
Okay, I have a quick question just to clarify.
Yes.
So the transfer RNA is carrying the anticodon?
It's not carrying the anticodon. If you notice, the anticodon is a part of the sequence. It is RNA. So if you look in here, let me use the highlighter here. As I mentioned, notice it's only a sequence of about 80 nucleic acids, all bound together. However, these 3 right here, because of their location, those are the special ones that are going to allow it to attach to messenger RNA using complementary base pair rules. So that 3 sequence of the sequence of RNAs is our anticodon.
So it's doing it just like 3 at a time? Is that?
Yes, well, because remember, the messenger RNA uses those codons, those sequences of 3 nucleic acids that are going to code for a specific amino acid.
Right.
So if AUG is going to code for a specific amino acid, if it's going to code for methionine, then I need a transfer RNA that is going to be able to bring methionine in to the party, because that's what the instructions are telling me to make. And so the way that I match this transfer RNA to the codon on the messenger RNA is by having the right anticodon in this location, so that while, if you think about it, there's going to be 64 different transfer RNAs, only one of them is going to be able to bind to this codon on the messenger RNA. And so that's how we make certain that only the correct amino acid is brought into the sequence.
Okay.
When we go through the process and talk about a process, it might help to make some sense when we see it that way. So we will do this together and see that, and hopefully it'll make more sense there.
So
Okay?
so the anticodon, it moves with the way that to build the amino acids, the protein, or is it always the same?
So
the right way.
I understand what you're asking. So the answer to this is to remember. So, okay, hold on. Let's do, because I made a mess of the board. Let's do this real simple. Now, if you think about it, there are 64 different possible combinations of transfer RNAs, right? because as we know, there are 3 sequences down here at the bottom and 4 of each of them that could possibly be. So again, one transfer RNA is going to be AAA. A different transfer RNA is going to be AAC. Another one is going to be AAU, and so on and so forth, right? There's going to be all these different possible anticodons, and every single one of them is going to be bound to a specific amino acid that it holds on to.
Okay.
So you have all these pieces carrying amino acids. Our goal is to put those amino acids together in the right sequence to make a functional protein. And the instructions for that are our messenger RNA. So our messenger RNA, let's say there's a sequence of our messenger RNA that was C, 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 and we'll put gaps in between the codons just to make it easier, right? A, C, A, G, G, C, and so on and so forth. What this first codon on our messenger RNA tells us is what the first amino acid we want is. So of the 64 possible transfer RNAs that are out there floating around in the cytosol, the only one that is going to be able to come in and bind to it is going to be the one that has the right codon. And so it would have to be the one that has GGG as its anticodon. That's going to be the only transfer RNA that will be able to come and sit on this particular location. And when it does that, it is going to bring its specific amino acid, and that's how we get the first right amino acid in. And for the next one, it's going to be UGU, and so on and so forth. So this is the instructions for oops, I didn't mean to do that. This is the instructions on the messenger RNA for making this. This is the sequence of amino acids we want, and it's the transfer RNA's job to bring the right amino acid to the party.
Okay.
And like I said, we'll actually talk about the process and see how this works, and it might make more sense then.
Okay.
Okay? But that's basically the job. The job of the transfer RNA is to bring the correct amino acid for the sequence. And so there's all these transfer RNAs holding on to specific amino acids, but only the one that matches our messenger RNA will be able to bind and start the process. Okay? And like I said, we'll see how that works. Before we can see how that works, though, we have to talk about our second or our third RNA, and that third RNA is ribosomal RNA. Now, what do you think ribosomal RNA helps us to do?
Move proteins?
True, but even more specifically than that, what do you think a ribosomal RNA does?
Make ribosomes.
Yeah, it helps us to form the ribosomes. Ribosomes are big, huge organelles, all right? And a ribosome is actually comprised of both ribosomal RNA and proteins. So both of those two things together, ribosomal RNA and proteins, make our ribosomes. Now, ribosomes actually are made up of 2 subunits. There is a small subunit. I'm going to draw this as a little bit of a box. Our small subunit. And we're not going to worry about what parts of it are DNA and what parts of it are ribosomal RNA, but it's the small subunit, and the small subunit has a specialization on it. This specialization on the small subunit is a little specialization right here where it is able to bind to messenger RNA. So our small subunit has a binding site that allows it to bind onto messenger RNA. Our large subunit has 3 binding sites. So our small subunit, let's say this, 1 binding site. Our large subunit has 2.5 binding sites. Oops, hold on. Do that. that, do that. Binding site 1, binding site 2, binding site and a half. Excellent. These 3 binding sites have special names. They are the A site, they are the P site, and they are the E site. The A site is where a new transfer RNA enters with a new amino acid. And based on that, what do you think A of A site stands for?
Amino acid.
Amino acid, there you go, excellent. The P site is where the ribosome holds the transfer RNA that contains the sequence of amino acids. And what do we call a sequence of amino acids again?
Protein.
Well, it becomes a protein, but at first it's a polypeptide. You're right, once it's fully functional, it'll be a protein, but when it's just a sequence of amino acids, it is a polypeptide. So guess what the P of the P site stands for? Polypeptide, exactly. And the E site, anyone know what the E might stand for?
Exit.
Exactly. This is where the transfer RNA exits the ribosome. That's why I commonly call it half a binding site, because the transfer RNA doesn't really stay there. It is just going to leave that site, and it's where the transfer RNA exits the ribosome, and so it is the E site. So there you go. That is our ribosome. 2 subunits, one small, one large. The small one has 1 binding site where it binds to the messenger RNA. The large subunit has 2.5 binding sites, really 2 binding sites, and then an exit site. An amino acid site where the new amino acid comes in, the P site where the polypeptide sequence is going to be held, and the exit site where our transfer RNA leaves the ribosome. Again, I've done a truly amazing job of drawing this. My drawing skills are amazing. Again, the job of the ribosome is to bring the messenger RNA and the transfer RNA together. We have those 2 binding sites in the E site. And again, if you didn't like my art, here is the pretty picture from your textbook that shows us what a ribosome looks like, where we have that large subunit with the A site, the P site, and the E site. And we have our small subunit with the 1 single binding site for our messenger RNA. All right, questions on that? All right, excellent. Translation is going to be one of our longest physiological processes. So like we've done before, we are going to go through this twice. I want to do it once where I'm just going to do it on the board as best as I can. Again, I don't have the whiteboard where it's easy to erase things, but I'll do the best I can to move these things around and make it make sense. For this first time, as I've said before, I strongly encourage you to just put your pens and pencils down, just watch, listen, learn. And then after we do that, we'll go through the entire process again with all the pretty pictures and all the pretty words and make sure that it makes sense, okay? But I also, because this is the longest physiological process, want to make sure we come at this fresh. So even though it's a teeny bit early, I think this is a good point to take our first break. So let's go ahead and take our first break here. It's essentially 9:10, so let's take a 15-minute break, come back at 9:25, and at 9:25, we will pick up from here. All right, so any questions on any of that? All right, I'm going to prep and get the board ready for us and so that we can go through this together, and then we will come back at 9:25, and at 9:25 we will pick up the lecture from there. All right, any questions? All right, see you in 15 minutes. Let's see if we can get this to work properly. All right, excellent. So, as I mentioned, we are going to go through the process of translation. And we're going to go through this step by step. So like I said, for the first time, I really encourage you to just sit back, watch, think, listen about the process. And then afterwards, you'll have the chance with all the pretty words and all the pretty pictures to be able to write to your heart's content. All right, so basically, what we have here on the board are 4 different things. We have our messenger RNA. We have our large subunit of the ribosome. We have the small subunit of our ribosome, and we have transfer RNAs. These 4 things are all going to come together, and they're all going to come together on a starting point. What would a convenient starting point be?
AUG?
Yeah, the start codon, absolutely. And so that is exactly what's going to happen. What's going to happen is first the small subunit is going to start working down the messenger RNA until it finds the start codon. And what was the start codon again?
AUG.
AUG. And lo and behold, right here is AUG. So it is going to bind to that messenger RNA at the start codon of AUG. Then our large subunit is going to come and bind on that start codon as well. And remember, it has 3 binding sites: the E site, the P site, and the A site. And the 4th thing that's going to happen is one of our transfer RNAs is going to come and bind at this start codon as well. Now, again, remember there are 64 different possible 64 possible transfer RNAs, but only one of them is going to be able to come in here and bind. And what is that one that's going to be able to come in here and bind? It's going to be the one that has the correct anticodon. And what would the correct anticodon be in this case?
UAC.
All right, excellent. So it would be U, A, C. And didn't make, so let's switch that. I think 36 is the right size. Perfect. There we go. That matches up. So it's got the right anticodon on it, so it's going to be able to come in and bind. And we also know it is going to be bringing an amino acid with it. And what particular amino acid is it going to bring with it? What does the start codon code for?
Methionine?
Right, methionine, excellent. And we can abbreviate it MET because it fits. Excellent. So there you go. Those 4 things, the messenger RNA, the large subunit of the ribosome, the small subunit of the ribosome, and the transfer RNA all come together, and they all come together on the start codon. Okay? So far, so good? All right. Now notice our A site is open. And because our A site is open, that means a second transfer RNA is going to be able to come in and bind in this site. Now, can all of our 64 transfer RNAs come and bind in this site?
No.
Only one, and that one has to have the specific anticodon. And what would we need that specific anticodon to be?
GCU?
Exactly, we would need it to be GCU. Only the transfer RNA that had the correct anticodon would be able to come in and bind at this site. And when it does, it is going to bring an amino acid with it. Now, do we happen to know what that amino acid is off the top of our heads? Because we have it memorized with
No.
CGA? No. So what we'll do is we'll just put AA2 in here. Oh, and that's way too big. However, if we really wanted to look it up on the chart, could we look it up if we cared? Yeah, we don't really care right now, but we could if we wanted to. Excellent. All right.
So just to clarify, the
Yes.
amino acid that the transfer RNA is bringing in corresponds to the messenger RNA that you started with?
Yes. So absolutely. So, but I will tell you that some charts will do it the other way. If you think about the charts, there are two ways you can think about the charts. The charts could have a messenger RNA codon that it would, and then, but it could also have the transfer RNA anticodon, and then it could also have the amino acid. So the good news is we only need to have one of these memorized. So as it turns out, we know that if there is the messenger RNA of AUG, that it is going to code for methionine. Right? But guess what? We also know what transfer RNA codon codes for methionine. What transfer RNA anticodon codes for methionine?
UAC.
UAC, exactly. UAC codes for methionine. So notice we could do it either way. If I gave you a chart that had transfer RNA anticodons and amino acids, would you be able to tell me what AA2 was here? If I gave you a codon and amino acid chart, would you be able to tell me what AA2 is? Yes. So notice either of those two charts would give us the correct answer. It just doesn't matter for the process. But we could, to be able to answer the question of what that second amino acid is, I would have to give you a chart, either a messenger RNA codon chart or a transfer RNA anticodon chart to be able to answer that question. But if you had one of those two types of charts, you would be able to answer that question. Okay? All right, excellent. So let's get rid of all of that. Perfect. All right. Notice we have filled up all of our sites as best we could. Obviously, we can't put anything in the exit site. The exit site's going to be able to exit. But we have our two transfer RNAs set up in here. And now that we have our two transfer RNAs set up here, it's time for the ribosome to do its work. And that ribosome does work in two ways. First, it makes a bond. Let's put these in order. One. Oh. It's going to make a bond. And then once it makes a bond, it is going to break a bond. Excellent. Those are the two pieces of work it has to do. Luckily, we know the process by which you make a bond. If we were to make a bond between two amino acids, what process would we use to make a covalent bond? Only I had learned how to make and break a bond when we were talking about, oh, I don't know, maybe carbohydrates. Way back in the chemistry lecture. I'll wait if you have to look back.
Dehydration synthesis.
There you go. We're going to use dehydration synthesis. We're going to use dehydration synthesis to make a bond. Excellent. And what process do we use to break bonds? Well, if we use dehydration synthesis to make the bond, what do we use? Oh, there we go, hydrolysis. We're going to use hydrolysis. Now, when we use dehydration synthesis, if you remember, this makes a water in that process. And what does hydrolysis involve? About using water to break a bond? When I get these blank stares looking back at me and it's 5 days till the exam, that always gets me a little bit nervous. So that is our goal. Our goal here is to make a bond and break a bond. Step 1. Let me make this a little bit teenier so that it fits in. No, not you. You, I want to stain there. Oh, that's too big. There we go. All right, excellent. So step 1, we are going to make a bond. And we're going to make that bond between the amino acids. So what happens here, I'm going to stick that in there. And let's use red. And we are going to form a bond between the 2 amino acids. Now, when we form that bond between the amino acids, as we know, a water is going to come out of that process. And I'm going to cheat and just make the water a blue circle. And we'll just know that that blue circle represents water. And the good news is, once we have that blue circle, that water can be used to break a bond. And what we are going to do is we are going to break that bond between the first transfer RNA and its amino acid. So we are going to first make a bond between the amino acids, and then 2, we break the bond between the first amino acid and its transfer RNA using the water from step 1. All right. So we made a bond and we've broken a bond. Questions on that? All right. At this point, our ribosome has done everything it can do in this location. And so what this ribosome is going to do is the same thing you do every Friday night when you get a little bored, right? When you get a little bored, you pick up your phone and you swipe right. And that's what's going to happen here. Our ribosome is going to swipe right. This whole ribosome is going to shift 1 space down the messenger RNA. Notice as it shifts down the messenger RNA, basically 3 things happen. Our first transfer RNA is now in the exit site. And is it being held to the amino acid anymore? No, because that bond was broken. So our transfer RNA is able to exit the exit site. And it leaves. Our second transfer RNA, which was in the A site, is now in the P site. And now that it is in the P site, it is holding on to the strand of amino acids, holding on to our polypeptide. And notice the third thing that has happened is that our A site is now open. And now that our A site is open, a new transfer RNA can come in and bind at this new location. However, can it be any old transfer RNA that comes into this specific site? No. It has to be the one specific for the anticodon with the specific right anticodon. And absolutely excellent, perfect. That anticodon is U. Whoops. I've got this too thick now, don't I? Oh, that's what's going on. I didn't realize I hit the bold. I don't care about you. It is UUA. Excellent. UUA. So only the transfer RNA that has the correct anticodon can come into this binding site. When it comes into this binding site, it is going to bring with it a new amino acid. Do we know what that new amino acid is off the top of our heads? Nope. Could we figure it out if we cared? Yep. Do we care right now? Nope. At least I don't. Because it doesn't matter. It's amino acid 3. Oh. comes into this spot. All right. Excellent. Guess what happens now? All our spots are filled, so what do we need to do next?
Swipe right.
Well, before we swipe right, we have to do some work. Right? Before you can move on to that next date, you got to deal with the first one. So we got to do our bonds. We got to make a bond and break a bond. Which do we do first?
Make a bond.
Make a bond. Where do we make that bond? Between the amino acids. Excellent. What process do we use to make that bond?
Dehydration synthesis.
And as a result of dehydration synthesis, what do we get from that process? We get a water. Excellent. Oops, nope, wrong button. Excellent. We get a water as a result of that process. I don't know why my water's changed colors, but it doesn't matter. Excellent. What do we use that water for? Break a bond. Allison, I see you mouthing the words, but you're muted, so I'm not hearing them. But I appreciate the enthusiasm. Excellent. We're going to use that. We are going to use that water to break a bond. Absolutely. And in this case, we break the bond. Whoops. We break the bond. Hold on. Let's go back. Right here between the second transfer RNA and the amino acid in the P site. Excellent. So we have made a bond using dehydration synthesis that has given us a water, and then we've used that water to break a bond between the transfer RNA and the amino acid in the P site. Now we've done all the work we can do at this location. So now what do we do next? Swipe right. There you go. Exactly. All right. So again, our large subunit swipes right. Our small subunit swipes right. And again, I can only grab them and move them one at a time. This is all happening. But we're going to the right though. So we're going right, swipe right and right. Excellent. When we swipe right, notice our transfer RNA in the exit site. Oh, forgot to move my letters. In the exit site now is able to exit, leaves. And again, once these leaves, there are enzymes that will go and put the correct amino acids back onto this. So it is actually possible. Some of these transfer RNAs can be used as many as 12 times during one translation of this messenger RNA. So there's nothing saying that it can't be used again. Remember, these sequences of amino acids are going to be hundreds of amino acids, in some cases thousands of amino acids long. So they can be used again, and they will go and get a new amino acid put on them, and then they could be used again. Our third transfer RNA is now in the P site, which again allows a new transfer RNA to come in. And that new transfer RNA, and let's go ahead and beat the dead horse to make sure we fully understand this process, is going to come in. And it can't be any old transfer RNA. It has to be the transfer RNA with the correct anticodon. And what would the correct anticodon here be?
GCG.
GCG. Excellent. And that is going to be bringing with it some specific amino acid. Excellent. Oops. Sure, I put a space there. Make sure it fits. Excellent. All right. All of our binding sites are open. So what happens next? Alfredo, maybe when we go through this again with the pretty pictures and the pretty words, it might help. Your book also has a great in focus, which does a job of describing this. I know doing this here on the board this way is a little bit awkward. Normally I'm able to draw this on the board, and I think to try to make it a little bit more sense. But if you notice, one of the things that's happening here is we're getting the same repetitive pattern of things happening again and again. The first thing that happens is that all of our components of the ribosomes settle onto one location of the messenger RNA. Then both our P sites and our A sites fill with amino acids. And that's where we're at right now in the process. Then, as people are pointing out, we then are going to do two chemical reactions. The first chemical reaction that takes place is we are going to make a bond using dehydration synthesis, and we make that bond between the two amino acids. So that is step one. We make a bond between the amino acids. Now, as we've pointed out, to make that bond with the amino acids, we use the process of dehydration synthesis. And that gives us a water as a result of this. And then we use that water to then break a bond. And that bond we are going to break using hydrolysis is between the transfer RNA and the amino acid in the P site. For the genetic codes, what you have to remember is we're using those complementary base pair rules. For the same way for DNA, RNA works the same way. Using hydrogen bonds, only cytosine can form a bond with guanine, only uracil can form a bond with adenine. So it's those hydrogen bonds that determine, or those complementary base pair rules that we talked about, that help to do this process. And again, it occurs again and again and again 100 times. Make a bond, break a bond, shift to the right. And again,
I have a question.
Yes, go ahead.
So on the small subunit, you said there's one binding site. Where is that binding to?
I didn't bother drawing it on this, but it's a great question. The answer to your question is that there is a binding site essentially here on the small binding site, which is what attaches it to the messenger RNA. So it is basically what is holding the messenger RNA and keeping the messenger RNA from moving around. Whereas the large subunit is then able to align the transfer RNAs with the messenger RNA. If the messenger RNA was able to move around, you might not get the correct sequences. Because if you think about this, if this shifted one space to the left, then the three codons, the three nucleotides you would read would be different. You'd add a different codon, get a different amino acid, and it would screw up your code. So basically, this is what anchors the messenger RNA in place so that it doesn't move around. Yeah, it's hard enough to draw these things and move it, so that's why I didn't bother putting it on there, but it is definitely there. Great question. Any others? All right, I'm going to cheat and go ahead and get rid of that because it meets my needs. All right, so we have made our two, we've done our two chemical reactions. We made our bond and are broken our bond. So again, it is time for this to shift. And again, as I mentioned, in a real messenger RNA, right, these sequences are hundreds, if not thousands of amino acids long. And so this process will continue 100 to 1,000 times until something different happens. And that something different is about to happen for us. So this shifts, I'll move that one out of the way for a second. This shifts and now something different has happened. What is that something different that has happened?
A stop
Stop
codon.
codon.
Exactly. Way back here, and let's cheat and emphasize this. This back here was our start codon, and this right here, start codon, and this right here is our stop codon. So we started here with a start codon, and this process continues till we reach that stop codon. Now, with a stop codon, remember our start codon also coded for methionine. But our stop codon, we said, doesn't code for an amino acid. So what's going to happen is a transfer RNA is going to come in. And again, can it be any transfer RNA? No, it has to be the specific one with the specific anticodon. And what would the specific anticodon here be? ACU, excellent. ACU, excellent, comes in. So now this very special transfer RNA with this specific anticodon is going to come in. But instead of bringing an amino acid with it, what this codon, what this transfer RNA does is it brings a water. The transfer RNAs for the stop codons do not bring amino acids. Whoa, that is huge. Let's do this again and do it smaller. Transfer RNAs for the stop codons do not bring amino acids. Instead, they bring water. Now, we can't use that water to form a bond with our fourth amino acid, but what could we use that water for? What can water be used for?
Break the bond.
Breaking bonds. And that's exactly what happens. This water is used to break the bond between our transfer RNA and the amino acid in the P site. Notice, once we break that bond, is there anything holding this together anymore? Before, there was a transfer RNA holding the sequence of amino acids. Is there anything holding the sequence of amino acids anymore? Right. Oh, I forgot to exit my E1. Let's get that one out of there. Perfect. Is there anything holding this sequence of amino acids in place anymore? No. And so what's going to happen is at this point, the entire structure disassembles. Excellent. So what's going to happen at this point is our small subunit is going to let go. Our large subunit is going to let go. Right? Our transfer RNAs are going to float away. Get rid of these things. And now we have our completed strand of amino acids. This completed strand of amino acids then travels to the Golgi apparatus, or Golgi complex, you can use either. And once it gets to the Golgi complex, it is modified into a functional protein. All right. Questions on this?
That new structure is called the tRNA, is that right?
What new structure? This thing that we just made?
Yes.
No, it's a sequence of amino acids. It's a polypeptide. We've made a polypeptide. Right? It's a sequence of amino acids. Yep, polypeptide is all one word. And again, that sequence of amino acids goes to the Golgi apparatus. What does the Golgi apparatus do to this sequence of amino acids? If only we'd learned about the Golgi apparatus in this class and what its functions might possibly be.
It modifies it.
Yeah, it's going to modify it. It's going to cut it. It's going to fold it. It's going to put the subunits together, and it is going to make a functional protein. And that's one of the last 2 points I want to make about this process. Notice every single polypeptide we make, what is the first amino acid we always use? Methionine, absolutely, methionine. Does that mean that if I were to reach into your body and pull out an amino acid, pull out a protein, every single protein in your body starts with the methionine amino acid on it? Turns out no. It is the case when we make the sequences, but remember, when this gets to the Golgi apparatus, one of the things the Golgi apparatus does is it cuts the top part of our sequence of amino acids up, and that's one of the things that allows it to fold up properly. So when we make it, they all have methionine as that first amino acid, but by the time it becomes a fully functional three-dimensional protein, that part has typically been cut off. All right? And now the second thing I want to point out, notice in this process, have we modified the messenger RNA at all? Have we changed the messenger RNA or damaged the messenger RNA? No, which means we can use it again. Because after all, if we're telling the nucleus we need to make proteins, do we typically just need one of those proteins? No, we typically want to make a bunch of them. And that's the nice thing about this messenger RNA. This messenger RNA can be read again and again and again and again. And sometimes we can read this single messenger RNA hundreds of times to make hundreds of sequences of amino acids to make a whole lot of the proteins. In fact, if you think about it, and I'll do this with a small box, because our ribosome moves down, it binds, and then as it binds, it starts to move. Is there anything stopping a second messenger RNA, a second ribosome from coming and binding and following it down the line? No, in fact, a single strand of messenger RNA can be being read by dozens of ribosomes all at the same time. Your book actually has an amazing picture that actually shows this. It has an illustration that shows it, but it actually has an electron microscopy view where you see a single strand of messenger RNA and you see like a dozen ribosomes all moving down it at the same time. So this messenger RNA will be read hundreds of times to make hundreds of these copies of amino acids to send them to the Golgi apparatus to make hundreds of proteins. All right? Questions on that? All right, we've done it here with my drawings, but like I said, let's look at the pretty pictures. Let's look at the pretty words from our textbook to make some sense of this and go through it all again. Now, this one isn't from your textbook. This is a very basic, simple illustration of this. Notice this one, they didn't even bother to put the exit site on it. They've just emphasized the two actual binding sites. And so, of course, the two actual binding sites would be the P site, and then this one over here would be the A site. So we have that here. But this simple illustration will still allow us to understand and see how this process is going to work. So the first thing that has to happen, and let me move this out of the way here. The first thing that has to happen is we have to have those four things come together. The four things that are going to come together are the small subunit of the ribosome, the large subunit of the ribosome, the messenger RNA, and our transfer RNA. Those four things need to come together, and they are all going to come together on one magical spot, and that one magical spot is the start codon. So we've got our transfer RNA binding to it, and again, not just any transfer RNA, but the one that has the specific anticodon that matches the codon. And it always carries methionine as the first amino acid. And so those four things come together. Oops. So again, step one of this process, four mystical, magical things all come together. Small subunit, large subunit, transfer RNA, messenger RNA, and they all come together on that start codon. And the other thing we know about that starting process is that first transfer RNA with the anticodon UAC always brings methionine as its first amino acid. All right? And that's just in the first binding site. We then have room for a second transfer RNA in the second binding site, the A site. Again, not just any old transfer RNA can come in, only the one with the specific anticodon for the codon. In this case, the codon is CCG, so the anticodon is GGC, and it brings some second amino acid. And again, using our table, we could figure out what it is, but we don't really care. Carries the next amino acid and brings it into this second space. At this point, all our binding sites are filled. And once our binding sites are filled, it's time to do work. And our work involves two chemical processes. We need to make a bond and we need to break a bond. So the first thing that is going to happen is we are going to form a bond between our two amino acids. We're going to use dehydration synthesis to do that, because that then gives us a water, and that water can then be used to break the bond between the first transfer RNA and its amino acid. So first we use dehydration synthesis to make a bond. We get a water, which we can then use for hydrolysis to break a bond. So again, it's a two-step process. Step one, make a bond. Step two, break a bond. Once we've done all this work, we've done everything we can do at this location. And so it's time to swipe right. Our whole ribosome shifts down the messenger RNA. Our first transfer RNA enters the exit site and exits. Our second one, which is holding the strand of amino acids, is now in the P site. And that opens our A site for a new transfer RNA to come in. Again, not just any transfer RNA can come in. It's got to have the correct anticodon, in this case, AGC, so that would be UCG. And it brings in the third. And at this point, the process continues. Make a bond, break a bond, shift to the right. Get a new transfer RNA. Make a bond, break a bond, shift to the right. Get a new transfer RNA. Again and again, hundreds, if not thousands of times to make a sequence of amino acids. And this process continues again and again and again and again until finally something special happens. What's that special thing that happens that changes the process? Yeah, we hit a stop codon. And what are the three possible stop codons again? See, many of you mouthing it, but many of you are silenced. So give me the start.
UGA, UAA, UAG.
Excellent. One of those three. And lo and behold, do we hit one of those next here? Yes, we do. We hit that stop codon. And when we hit that stop codon, remember what the stop codon does is that stop codon brings a water. And when it brings that water, that allows us to break the last bond. And once we break that last bond, everything kind of falls apart. We have our completed sequence of amino acids, our completed polypeptide. Our ribosome disassembles, and that messenger RNA is completely unchanged, so it can be read and used again. Now, as I mentioned, this illustration that we've been looking at here with the pretty pictures are fairly simplistic, But as I also mentioned, your book has this amazing in focus where it does an amazing job of showing this process, where again, it shows the messenger RNA leaving the nucleus, binding to the 2 units of the ribosome, our transfer RNA coming in. It shows the step-by-step process of all of this going on. And notice it even shows the enzyme that puts the amino acids on the transfer RNAs. So here we have this great illustration. So again, if I know a couple of you have mentioned that you're struggling with this a little bit. If the way I've described it and my words aren't helping, I strongly encourage you to read this in the textbook, because your textbook does an excellent job of describing this as well. And sometimes hearing it in a different voice or reading it in a different voice can help you to understand these processes better.
Quick question.
Yes.
So the process of the codon, I don't know if I'm going to say this right, but the codon coming in and making the peptide bond and then moving on to the next one, that is the elongation process?
Yes, so elongation in this case is where the transfer RNA brings in a new amino acid. It is then, and then be careful, the ribosome is actually what's doing the work. The ribosome is what forms the bond between the amino acids and breaks the bond with the old transfer RNA. So the ribosome is the one doing the work. So basically elongation is this process of continuing and then sliding to the right, where we can then bring a new one in. So this is that process of continuing to add more and more amino acids to the sequence. All right, so notice, if you think about it, yeah, this basically has the same 3 processes as transcription, right? Initiation, where it binds to it, gets our starting point, right? Elongation, where it's adding something to it, and then termination, where it ends. But aside from the names being the same, the processes themselves are dramatically different. All right. Here's that simple illustration, as I mentioned, where we see the ribosomes binding to the messenger RNA, sliding down to the right and coming off, so we can be making multiple sequences of polypeptides at the same time. And again, the drawing is pretty simple, but here is that great electron microscopy view. So here with an electron microscope, we're able to see a single strand of messenger RNA. We can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, right? Over a dozen different ribosomes that are bound to this, all sliding down that messenger RNA, all making strands of polypeptides all at the same time. All right. Lastly, this is a great summary table that shows us this process of how this works. Right? We go from that template strand of DNA to messenger RNA to codons of messenger RNA to anticodons on our transfer RNA that code for an amino acid. So here in that nice, we see all of that view, all of that as we go down. And again, we can think of it as, I'll show you another picture of this in the same time. All right, we have described these processes, but is this something that you should be able to do yourself? Can you go from template strand of DNA to a codon to an anticodon? Should you be able to make that process and do that process? Absolutely. Now, the last step of that, figuring out what amino acid it would be, would you need a table to be able to do that? Yes. But if I gave you that table, should you be able to go from a strand of DNA to a sequence of amino acids? Absolutely. We do have one issue though, right? Because as we mentioned, we have our DNA inside of our nucleus that contains tens of thousands of genes. And those genes code to make proteins. So our eyeball makes eyeball proteins, our toenail makes toenail proteins, our liver makes liver proteins. Those 3 cells and all the other cells in our body make different proteins because they have different DNA. Your toenail cells, your liver cells, and your eyeball cells have the same DNA or different DNA? Same. Laura's got it. Again, I saw her mouthing it. Absolutely. Same DNA. Absolutely. The same DNA. So if they have the same DNA, why are those 3 different cells making different proteins? Remember, we have this issue. only 1.5% of our DNA codes for genes. So as we said, as we throw that dart, the chances of us hitting a gene is very, very small, let alone hitting the right gene that we need to make the right protein. So if every cell in your body has the same DNA, how do we tell toenail cells to make toenail proteins, eyeball cells to make eyeball protein, liver cells to make liver proteins? How do we do all of that? No, we have the same DNA, we have the same RNA, we have the same transfer RNA. How do we tell different cells to make different proteins and when to make those proteins?
Is it different base sequence? No, wait.
All have the same base, they all have the same genetic material inside. So how is it possible that we could tell a cell, one cell to make protein A, another cell to make protein B? And here's the other thing. Do we necessarily want cell A making protein 1 all the time? No. So not only do we need to tell it which specific protein to make, but we need to tell it when to make that protein. If only there was a way we had for communicating to cells. Oh, wait, I think we have 2 organ systems involved with communicating to cells. What were those 2 organ systems again? Endocrine system and the nervous system. Excellent. Which one do you think we use to tell cells which proteins to make? Does every single cell in your body have a nerve that goes to it that tells it what to do? No, exactly, endocrine. Hormones. The primary way we tell cells which hormones to make, which proteins to make and when to make them is hormones. Hormones and other chemical signals is how we tell cells which proteins to make and when to make them. So this whole process is regulated by hormones. Here we go. So again, you should be able to do this as well. Go from template strand to messenger RNA to transfer RNA to amino acid, as long as I gave you the last half of this, right? So as long as I gave you something like that, you should be able to do that work. All right, questions on any of that? Excellent. All right, we are doing great on time. This is perfect. So this is—
I have a question.
Yes.
I mean, just to reiterate, I think you just answered it. I just want to be sure.
Sure.
So in terms of transcription translation, like the test example is be able to take that initial strand of DNA and then go through the replication process into making the polypeptide chains.
Sure, absolutely. So again, think of it in 2 ways. I could ask you to describe the process of translation. And if I ask you to describe the process of translation, you kind of would do what we did here on the whiteboard, right? Describing this process, right? Where we talked about how, it binds using the anticodon. And technically for that, would you even necessarily need to know what the strand of messenger RNA was to be able to describe it? No. All right, so you wouldn't have to necessarily do something like that. However, again, because you describe it, you would just say using the complementary base pair rules, you would bind the transfer RNA that would be the right one to match it, and yammer, that you would go through it that way, right? However, let's go back, let's do that. So I can do that. At the same time, could I give you a strand of template DNA? So if I gave you a strand of template DNA, and I told you that strand of template DNA was ACT, CGA, TT, CJ, GCT, right, like that. Could I then, based on that, have you do all the work? Could I have you tell me what would be the messenger RNA that would be produced as a result of this? Once you did that messenger RNA as a result of that, could you then tell me what the transfer RNA anticodons would be that would match up with those? And then once you did that, if I gave you a table, could you give me which specific amino acids would be necessary? And again, we'll put an asterisk next to that because I would have to give you some table for that. Absolutely, you could do that. And in fact, should you also be able to tell me what the coding strand of DNA that is complementary to it would be? So could you actually do the work of being able to do all of this? Absolutely. So both of those are potential essay questions. Could I give you a strand and make you do all this work? Yes. Could I have you generically describe this work? Yes, absolutely. I guarantee those are 2 possible essay questions on this exam. Absolutely.
Will we be able to have note paper or scratch paper to work this out?
So remember, you have that whiteboard. Remember, part of that syllabus quiz was that where you had to draw the blue star on that. That whiteboard is your scratch paper. I can't have you have physical scratch paper with you because it's too difficult, right? Yes, oh, you could show me the page, but who's to say someone doesn't switch it out? No, you can't have any physical scratch paper here, but you have that whiteboard, which is something that you could bring up and use as scratch paper. And remember also, when you have your space where you're writing your essay, if you want to use the top part of that, because again, there's no limit on space for that. If you want to mind dump all your information there and then draw a line and say, ignore everything above this, and then just write your answer underneath that, that would be perfectly acceptable as well. So any of that is fine. I want you to be able to mind dump this information whichever way is comfortable for you. Well, so again, you don't have to use the whiteboard. You can use the part where you're writing the answers. If you want to use the top part as your scratch paper, and then you don't even have to erase it. You can just say, ignore everything above this, and then just write your answer under that, and I will do that way. But I can't allow you to have any kind of physical scratch paper or a whiteboard or anything like that that you can use. Because I actually had a student who did that. She was sitting on her couch doing it, and her desk, her lap desk was also a whiteboard. So she was using that to write her notes and stuff on. And she showed it to me that it was clear and then wrote on it and stuff like that. But even that, you can't do that. So there can't be any physical kind of scratch paper or whiteboard or anything else that you're using. But use the resources that you do have in front of you to be able to do that. Okay? All right. Any other questions on this process of translation or transcription, this process of protein synthesis? How many possible essay questions did I say you could have based on this? 3. What were they again? Describe each one and compare them. Excellent. One of the important things in a compare and contrast type of question is that when you are comparing and contrasting them, I want you to actually compare and contrast them. What that means is I don't want you to describe all of process 1 and all of process 2. What that also means is I don't want you to give me random descriptions about those things, right? If you were describing apples and oranges to me, and you said that apples were either red or green and oranges are sweet, is that a comparison? No. Now, I do not care how you organize this information. But one way, if you struggle with these types of questions, that may sometimes make it easier for you to organize this compare and contrast, is to basically do it in 3 columns, right? Let's say our job was to compare differences between translation and transcription. We can do that first by saying, what is the characteristic that we are comparing? And then we can do that for translation, and we can do that for transcription. Oops. So someone tell me one of the ways these things are different from each other. Someone tell me one way translation and transcription are different from each other. Okay, location where they occur. Where does translation occur? Nucleus. Where does transcription occur? Wait, no, that's not right. Where does translation occur? Yeah, transcription occurs in the nucleus. Where does translation occur? Rough endoplasmic reticulum. And since Alina wrote it out, I will cheat and abbreviate. Excellent. Give me another way they're different. Placement. What do you mean by meaning what happens first? Adina, can you give me an example of what you mean by that?
Could that be a point that we say that transcription has to happen first and then translation?
Oh, I guess technically the order
Because
that they occur is a different
initiation and stuff, so that couldn't be a point.
Well, so I actually like that better. So if you think of it in that case, if you think of it in case of, like, for instance, what is the end product of the process, right? The end product of the process of transcription is that messenger RNA, right? So we get that messenger RNA, and you're right, messenger RNA is then used for translation. And what is the end product of translation?
Polypeptide chain.
Yeah, exactly, a polypeptide. Excellent, right? I guess technically the order that they occur is kind of a difference, right? Because obviously this one happens first and this one happens second, but it really isn't very descriptive of the two processes. I guess begrudgingly, I would give you that, right? This is a perfect example. I don't know if I've mentioned this in this class yet. I'll probably do it when we talk about the exam. Often on the exam, and it's a little harder because I don't get to physically write on these, but I do try to make notes on them when on the exam so that when you get a chance to see them. Oops, no, hold on. I don't want that. Ah, good gravy. No. I hate the whiteboard. All right. Excellent. Black. On the exam, sometimes you will see me write the words or the letters. Okay. If you see me write okay, that is my way of telling you that you got the points for it, but it isn't 100% correct. What I find is when students look at their exams, they do not pay any attention to the things that they got right, and they solely focus on the things they got wrong. So if you get full credit for something, you assume you got it right and you don't pay any attention. If you see me write okay for one of your answers, what that should tell you is that I am giving you full credit for this because I understand where you're coming from, and technically it may be correct, but it really isn't the kind of answer I'm looking for, and that's probably what you would get here. This isn't really a good way of comparing the two of them. Technically, I think it's accurate, so I wouldn't mark you off for it, but there are better ways and more descriptive ways. And since this is a cumulative class where you need to know this information at the end, this is my way of saying to you that this could have been better, or this needed a little bit more. You got full credit, but be aware this isn't 100% accurate. Okay, does that make sense? So you may see that sometimes, and I will write that there. All right, give me at least one more. What's one more big difference between translation and transcription?
Transcription uses the same language and translation changes the language.
I like that. Excellent. In transcription, we stay with the same language, right? From nucleic acid to nucleic acids. And you're absolutely right. In translation, we change languages from nucleic acids to amino acids, right? Now, again, as I mentioned, you don't have to line the information up this way. You can write them out as sentences, you can write them out as paragraphs, but like I said, if you have problems with these types of questions, then I think actually taking the time to figure out what characteristic you're comparing really makes it a comparison and makes it easier to make sure you're comparing apples to apples and oranges to oranges. Could we add the core idea of each process? We can say that transcription is where information is copied, whereas translation is the change of language of the information. True, but again, if you're going to talk about language, then be more specific about language. It's copied in the same language, whereas translation, we're changing the language of the information. So I think that would be the only thing that I would add to that. All right.
Did you say, I don't think I followed, did you say that setting up the answer this way would be okay, or does it need to be like typed out paragraph format?
Great question. I do not care about the format of your answers. Not just this type of answer, but any answer. I do not care about the format. What I care about is that you have the correct information and you have the specificity and you have the details. So if this helps you think better, great, use this. If writing out sentences helps you think better or paragraphs help you think better, I don't care about your format as long as you have the information. The one caveat that I would add to that is the one trap that some people fall into. If they use a table like this, or if they use bullet points, sometimes they're too brief in their descriptions. And that will cost you points. To maximize your points in this class, as I normally say during the first exam, I usually use a pen and a half of red ink writing out explain or describe or be more specific or things along those times. The way you maximize your points in this class is by being specific, giving me details. Again, here's a perfect example. If you just said that in translation, you change the language and in transcription, the language is the same, that information is correct. But if instead you said that transcription, we use the same language going from nucleic acids to nucleic acids, whereas in transcription, we go from nucleic acids to amino acids, notice that second one has more detail. The first one wasn't wrong, it just lacked specificity, it lacked detail. And that subtle difference is enough to cost you points. So I want specificity, I want details. So as long as you give me the details, as long as you give me that specificity, I don't care how you write it out. Okay? Excellent. All right. And you don't even need these characteristics. If you just tell me that translation occurs in the rough endoplasmic reticulum and the transcription occurs in the nucleus, that is a comparison. You don't have to say they're different in where they're located, right? So you don't even have to give me the characteristics if you don't want. For me, it's an easy way to organize my thought process to make sure that I'm really comparing apples to apples and oranges to oranges. Okay? All right, excellent. Any other questions on this process? All right, so here is what we are going to do. I am going to, how do I want to do this? Where are we time-wise? We are doing good on time. All right, here's the plan. I am going to unlock your practice lab exam now, okay? This practice lab exam, as I've mentioned before, is all questions that have been on previous exams, so this is a good gauge of the type of questions I will ask on the exam. Let me take a peek at it to see what we're looking at here. It is 20 questions, and you will have, I believe, 20 minutes. No, I give you 25 minutes. All right, so, oh, and it does use Proctorio. Ah, it is using in Proctorio. Excellent. Okay, I forgot I had set that up. All right, perfect, even better. This is a great way to get your Proctorio practice in. 20 questions, 25 minutes, normal Proctorio rules. This is exactly how the lab exam is going to run. All of these questions have been previous questions on the exam. So what you are going to be required to do is to take these, answer these questions. I think, and actually I think that's where it's different. I think in the setting up of this, while it is, I'm using Proctorio, I don't think I set up the video for it. Let me see. Yeah, so this one, excellent. Okay, so this one doesn't have the video set up for it, but it uses all the other lockdown things of, all of the other lockdown components of the exam. So again, this is locks down your tabs, locks down your other things, but you don't have to worry. No, you don't have to worry about clearing your workstation. You don't have to worry about being video recorded and scanning your room or anything along those lines, because this test doesn't count. It's for 0 points. This is a way for you to gauge the information. What I'll say is all these are questions that have been on previous exams when we did this in the classroom. So it is a good gauge of both how I ask questions and what type of questions I'm going to answer. You have 25 minutes to take it, but what we're going to do is I'm going to give you 40 minutes of time, so that way you can also get a 15-minute break. So if you want to take your break first and then take the quiz, you can do that. If you want to take the quiz first and then quiz first and then break's fine. If you want to take a break first and then the quiz, that is fine. But what we will do is we will meet back here in 40 minutes. So that will be at 11:10. At 11:10, we will go over the exam together, talk about the exam together, talk about the process of the exam and everything that goes over that, and make sure you guys are prepared for the real thing on Monday. All right, at that point also, if you have any other questions about any of the material we covered, I will answer those questions at that time as well. All right, so questions on any of this? All right, so here's what's going to happen. I'm going to set up the quiz. It is going to be available until, like I said, we'll make it 11:11. So, oops, hold on, 11:11. AM, available that way.
Professor, I have a quick question.
Yes.
So for my chemistry quiz, you left a comment that my camera and my audio, it was working poorly. Would you be able to see on this exam that we're going to take, not this exam, sorry, the practice exam, would you be able to see if it works properly? Is that set up?
No, unfortunately, because this isn't going to record your video on that. But did you take the chemistry quiz and the, I'm sorry?
I did take the chemistry quiz.
And did I mention that on both of them?
So on the syllabus quiz, there was no comment, but on the chemistry quiz, there was. So I was I just want to make sure that's all working.
Well, what I can do is what I would recommend is we can talk after class. If you want to come to my office hours, I can reset something up for you so that you can test it again and make sure it works properly that way.
Okay, thank you.
Okay. All right, so any other questions on this process?
Where do we find the quiz?
The quiz is going to, I haven't posted it yet, so it's going to be in the quizzes where everything else is. Alfredo, no, you cannot take these on the phone. You have to have a computer. You have to have the, actually without the video, I don't know if it can be taken on the quiz. I don't know if it can be taken on your phone or not, but the real exam you need to take on the computer. So I would recommend you do it that way. I think with the other lockdown things, I don't think it can be taken on your phone. So all of these things need to be taken. These quizzes need to be taken. And in fact, I think I sent you an email about that with your other exams. So if you have not responded to that, please take a look at those. Adina, anytime is fine. I'll respond to them as I can. All right, so any other questions on the quiz itself? All right, so take a break, take a quiz. Let's meet back here at 11:10. Let me post that for you guys now. There you go. And again, by using Proctorio, it will input the access code for you. So you don't have to worry about the access code. Proctorio will input that for you. All right, that is available. Meet you guys back here in, like I said, we'll at 11:10. So at 11:10, I'll meet you guys back here. All right, good luck. Have fun. Again, this doesn't count, but treat it like a real exam. Try not to look at your notes. Try not to look at other things. Try to do your best on that. And I will meet you back here at 11:10. So for the, so again, I haven't finalized your exam yet, so I can't give you the actual numbers, or if I had finalized it, I would give you the actual numbers. But here's basically the case. The lecture exam is usually somewhere between 20 and 30 multiple choice questions, somewhere between 2 and 6 fill in the blank, and usually somewhere around 7 to 9 essay questions. So depending on the size of it, typically you get somewhere between 2 hours and 2.5 hours for the lecture exam. Again, you're not required to use all that time, but that is the time that is available to you. This lab exam, as I said, is probably somewhere between 60 and 70 questions for this lab exam, somewhere in that range. And so typically I figure about a minute per question for the lab exams, but there is the technical things like the scanning of the room and all the kind of things that go along with that. So usually I do 10 to 15 minutes more. So usually the lab exams have somewhere like 75 to 90 minutes. So worst case scenario with a really big lab exam, which this one won't be, when we get to the bones, when we get to the muscles, those will have 80 plus questions on them. But so the most you'll have about 90 minutes, an hour and a half for the lab exam. and about 2.5 hours for the lecture exam at the most. So if you think about it, that's 4 hours for the exams. And 4.5 hours for the class. So you can take a break in between. I encourage you to take a break in between. Also, I will tell you from the past semester and a half of teaching this class, where most people have problems is the lab exams. Some people, especially if they tend to have older computers, the loading of 75 high image graphics can chug on the computer a little bit. So most people who've had problems have problems on the lab exam. So I encourage you to take the lab exam first, so that if you have problems, right, if you have a spectacular, if you have a gaming computer that you're using, then take them whatever order you want, it's going to be fine. But if you have a 5-year-old laptop, you might want to consider taking the lab exam first to make sure it functions properly, so that if there's any problems, you can get that worked out. And again, I think I've made a point of emphasizing this, but I'll emphasize it again. If you get kicked out of the exam, or if the exam locks up or something along those lines, your job is to contact the Proctorio Tech Support. The Proctorio Tech Support are the ones that can resolve your issue and get you back into the exam. I do not have the ability to get you back into the exam. So if you get kicked out of the exam, don't write me an email asking me what to do. Contact the Proctorio Tech Support, which is right there on the page, and they can get you back into the exam. And remember, the clock keeps running. If you get kicked out of the lab exam, the clock keeps running on that. So if it takes you, if you decide to go and have lunch and come back and then try to log on, then you're going to miss that half an hour or 40 minutes out of the time that you have. So the clock keeps running while you get kicked out. So don't get kicked out so that you can take a break and study or something like that, because it's going to count against your time. All right. Yes, Laura, the lab exam, like this practice exam, is primarily identification type questions. But could I ask you the function of something? Could I ask you to identify that rough endoplasmic reticulum? Or could I ask you what the function of that rough endoplasmic reticulum is? Absolutely. So there can be some functions. Again, remember, this is one class. It's not like lab class structure, lecture class function. It's all one class. They're just different modalities for learning the material. All right. So I'm happy to continue answering questions on the lab exam, but at this point, it's also open for any review questions. So if you guys have any questions or any material you're not clear about, this is a perfect opportunity to ask that, because as someone pointed out, you guys have your exam in 5 days. So if there are things that you're struggling with, now's the time to ask those questions to make sure that we can come up with the right answers to help you guys to be successful on the exam. Remember, as I mentioned, if you guys don't ask questions, then I assume that you've all mastered the material and I make the test harder. Let's see, for chemistry chapter 2, we only need to know what we went over in class. Yes, and again, that's a good general rule for everything. As I've said before, my job in this class is to be your tour guide, to show you the things that are important, and the things that are important are what's going to be on the exam. If there is a concept that we haven't talked about in class, that you haven't had a homework assignment about, right, that isn't been addressed in lecture, it's not going to be on the test. In very rare instances, and I'll be honest, the next section, because of the way the holidays fell out, we're going to do what's usually 3.5 days worth of lecture, we're cramming into 3 days. So if we're running really late on the skin, one of the things I may say is, look, I don't have time to talk about burns in class today. So make sure you read that in the textbook, because it's going to be on the exam. Right there, I'm specifically giving you a piece of information that you need to go study on your own. But if I don't do that, if we don't talk about burns in class, if we don't have any homework assignments on bones, we don't get to it in lecture, it's not going to be on the exam. So yeah, so in general, the best rule is to focus on the things we talk about in class, but especially for chemistry. The whole first part of that chapter, like I said, is the stuff you should know coming into it. What we talked about is the macromolecules. So that's the part of chapter 2 that I will hold you responsible for. No, the second to the last slide of the exam was not the left femoral, it was the left inguinal region, or iliac. Both of those are acceptable terms. It was basically right at the hip, right kind of above the hip. So femoral is really the lower part, the upper part of the leg. Got it? No more questions?
Professor, I have a question.
Yes.
Would you explain what the poles are again during the during mitosis again? I believe it happens during early prophase, like what the poles are and let's see.
So yes.
Or just, yeah, just the poles right now.
Yeah, no problem. So yeah, absolutely. So, and also to real quickly answer Haley's, yes, anything that you have homework or the lab simulators, anything you've done on the Labsters or the PhysioX is also material you're responsible for on the exam as well. Yes. So the same way that the Earth has poles, a north and a south pole, a cell does as well. Now, again, it isn't like there's some striped pole up at the top of it that signifies where it is. It just happens to be a region of the cell, and it could be anywhere. Why they're significant, as we talked about, is that those centrioles, there's that one that way, and one this way, and another one that way, and another one this way. Those centrioles during prophase, as you're correct, in early and late prophase. In early prophase, they start to migrate where they go to the opposite sides of the cells from each other. And by late prophase, they've actually made it. So by late prophase, one of them is on one side of the cell, and the other of them is on the completely opposite side of the cell from it. And that's what we consider the poles. All right, and so those poles are the starting point for those spindle fibers that we know that are going to come out from them. The other reason the poles are significant is because we also know that our chromosomes in metaphase is going to line up on that metaphase plate, which is basically the equator of the cell, the midline between the two poles. So that's why the poles are important. The poles are where the centrioles go. And the equator, the metaphase plate, is the line basically between them where our chromosomes are going to line up during metaphase. But it isn't like there's a physical structure at the pole. It's just the 2 points that the centrioles go to. Okay?
Oh, yes, that makes sense. Thank you.
All right. Next question.
In PhysioX, we did a lot of diffusion and filtration, just kind of in general.
Yep.
Do we need to know about diffusion and filtration in general and osmosis, or more how it relates to the cell membrane?
Both, right? As we've talked about, we spent a lot of time in lecture talking about the different mechanisms for getting across a membrane, both passive and active, right? We talked about those, the differences on the passive between simple diffusion versus channels versus carriers. On the active, we talked about the primary and the secondary, those co-transporters, the antiporters and the symporters. So absolutely, we need to understand the mechanisms by which things get across the membrane. But do we also need to understand the practical uses of those, like we did in the PhysioX, where you actually see the diffusion, where you see the transportation taking place? So could you have to describe, right, that type of a simulation, or describe what would happen if I gave you certain conditions and how that would work, or something along those lines? Absolutely. So you need to understand the general concepts of it, but also be able to use the information practically as well. And the fact that we talked about it in lecture, and that you did have a big, huge lab assignment that we did it for the PhysioX, means that it's probably a pretty big concept. And when you've got a pretty big concept, especially when we've studied it by different modalities, is there very likely to be an essay question on that on the exam? Yeah, I don't think I'm giving away any secrets by saying, yeah, definitely membrane transport is going to be a huge concept for this. Protein synthesis was a huge concept we covered. Mitosis was a huge concept. Again, as sophisticated students, you guys should be able to go through the lectures, go through the materials that we've covered, and pull out the big concepts that there's going to be potential essay questions on. Again, my goal isn't to be tricky. I tell you the things that are important. Those things that are important are the things that show up on the exam. Can we only access the information from Labster by opening the virtual lab, or is there a web page that summarizes them? That's actually a great question. I know in Canvas, you guys have that Labster dashboard. Once you complete an activity, you can go and see your score. Since I'm the instructor, I'm not sure what your guys' view is. I don't know if you click on that, if it shows you the information. But if it doesn't, then yeah, you can always go back and do the activity again. Okay, it doesn't. So yeah, what I see as the instructor is different from what you guys see on that dashboard, so I didn't know that. So yeah, if it doesn't, then it looks like the only way you would be able to get that Labster information again is by going through it a second time. But again, like the Physio X's, you can go through these things as many times as you want. Any other questions? It's noon. We finished a little bit early. It shouldn't affect your score. If it does, because again, I see what your top score is, and then like I said, I've manually changed them to full credit. I don't think it should change your score. If you do it again and it does change your score, email me and I'll fix it. Because yeah, it shouldn't affect your score, because your score should be based on whatever your top one is. And then when I manually change it. But on the off chance if it does change, something wonky happens, let me know and I'll fix it. All righty, anything else? All right, so here are the 3 things that I will remind you. I have office hours right after class today. I will be closing this window and switching over to my regular office for that, so that we compile the video for this. So if you have more questions that you think of, I'm there for the next hour or so. If this time I'm not available for you, again, I respond very good at responding to emails, especially with a test on Monday. I don't have any big plans this weekend, so I'll be around and be able to respond to emails if you have questions that way. Lastly, I want to remind you on Monday, even though if you look in Confer Zoom, it shows that there is a lecture. There is no lecture on Monday. Monday, you are just taking the exams, lab and lecture exam. I will be available by email if you have any questions, and then if anybody has any problems, I can try to resolve it. But like I said, you want to go to Proctorio first. Their tech support are the ones that can get you back into the exams. I do not have the means of doing that. So if something wonky is going on, let me know. Like today with the practice exam, even if you've set up your Proctorio, sometimes when that occurs, for whatever reason, the Proctorio unsets itself. I don't know what the right term is for it, but it disconnects. And the way to tell is by looking on that left-hand tab. If you see it say secure Proctor exam, then that means that the Proctorio is not active. Click that link, it will activate Proctorio. And then once you go in, you should be able to take the exam. If you try to take the exam and it asks you for the access code, then that means your Proctorio is not set up properly. All right. And unlike this practice exam, you will need your camera, you will need your microphone for this. This is the full thing, like the chemistry quiz, like the Proctorio syllabus quiz. All right. Read the questions carefully. People often lose points, not because they don't know the information, but because they don't read the questions carefully. Read the questions carefully so you can answer them correctly. You have plenty of time to take these exams. So take your time, read them carefully, answer them correctly, maximize your points. All right. Any questions? All right. Good luck on Monday's exam, and I will see you in lecture bright and early Wednesday morning. Have a good weekend.